Thanks, Vanessa, and thanks everyone for sticking around for the graveyard slot of the day. So I appreciate everyone's persistence. So I'm just going to talk a little bit today about biobanking initiatives in Ireland, particularly what's involved, what they can achieve, and then hopes for the future. So I think we're all well established now. We know that Alzheimer's disease and other dementias is a looming both personal and societal crisis, with, if current estimates persist, 1 in 3 of us in this room developing dementia, and 1 in 2 of us will care for somebody living with dementia. And there's been a real amount of progress back from 1906 when this was first described through to the 1990s when the amyloid plaque was discovered, amyloid plaque and APP gene was discovered. But a real step forward that has changed things has been the discovery of biomarkers, be they CSF biomarkers of Alzheimer's disease or amyloid PET scans. And really that had a transformative effect, particularly for research and clinical trials. You're probably familiar with the narrative that we've all heard up until very recently of, Alzheimer's disease isn't treatable, but actually if we look back at all those failed clinical trials, 1/3 of people didn't even have Alzheimer's disease. So really it was biomarkers that have transformed the research landscape, because we've now able to clinically, biologically diagnose people and make sure that people are getting an accurate diagnosis. However, we've heard a lot today about equity, and really a barrier to biological diagnosis has been the cost associated with them and the scalability. CSF is required via lumbar puncture, so you need to have specialist centres, and amyloid PET scanning is both costly, has radiation exposure, and has infrastructure requirements. Whereas blood biomarkers really represent something of a holy grail. They're cheap, they're accessible, they're easily repeatable, and have the potential to democratise a biological diagnosis of Alzheimer's disease. So why are they important? So they increase diagnostic certainty. So even in the top specialist centres, around 1 in 3 people with a clinical diagnosis of Alzheimer's disease are misdiagnosed at post-mortem. And as Marwa has outlined, an accurate diagnosis means you get the appropriate supports and the appropriate treatment. So it's really important that we have biological confirmation in individuals, because it can shape treatment. It also aids prognostication and long-term care planning, and this is some nice data from Holland, where we see people who had a biomarker-supported diagnosis actually did better. So those with a biomarker diagnosis and without, and you see there's greater survival time and delayed time to institutionalization. So having a biomarker in itself, even without access to any disease-modifying treatments, it has an impact. But as we all know, the landscape is changing, and now it can guide therapeutic decisions, which is particularly important as we enter an era of disease-modifying therapies. And really, this is the proof of the importance of amyloid, with anti-amyloid treatments that remove amyloid from the brain have been shown to slow disease progression. To have these agents, people need to have a biological diagnosis of Alzheimer's disease. So what's the Irish landscape like? So this is a position paper from a working group that was published in the IMJ, and it really reflects current practice in Ireland, where we're very much of a pathway of CSF being our main diagnostic tool, with amyloid PET only reserved for certain cases where people can't access lumbar punctures, such as they're on anticoagulation or other things like that, and that we would rely on the use of amyloid and phospho-tau or ratios. So why a biobank? So this is something that we wrestled with in Tallaght. We were doing, well over 200 CSFs a year, but there was just a diagnostic sample. People were very keen to participate in research, but we didn't have an outlet. And what we actually tried to do was then set up a biobank so that people at the time of their diagnostic CSF sample could donate a sample for research. And this has led to a really valuable data set. So we have biological samples linked to clinical, cognitive, and imaging data. And it supports then research into not only Alzheimer's disease, but those other conditions that also cause dementia, like Lewy body disease, vascular cognitive impairment, and other related disorders. And something that's particularly been useful for is looking at that field of moving forward and getting blood biomarkers. And I'll talk about that in a little bit more detail. And this is just our protocol paper, which outlines the process we went through to set up a biobank. And this was work that was really led by Adam Dyer at Tallaght. So what we collect, so basically what we collect is the routine clinical data. So what this isn't is it's not an extra burden on the workflow. So our routine clinical assessments, the routine ACE is recorded. We record if people have neural imaging and if they're on treatments, and then we collect biological samples. So we collect plasma, serum, DNA. For a subset, we're going to collect PAX gene tubes and dried blood spots, and we also collect CSF. And what can it achieve? I guess that's the big thing. What's the point of collecting all this data? What's the point of collecting the samples? What can happen next? So this is some work that we've been able to carry out due to having a biobank. So we looked at the real-world performance of this blood biomarker phospho-tau217. And phospho-217 is a blood biomarker of Alzheimer's disease that has performed really well in research studies. But as we know, some research studies, they're very much, tightly controlled cohorts with eligibility criteria. And what you want to see is what happens in the real messy clinical world where people have chronic kidney disease, or people have a different range of BMIs and a different range of comorbidities. So we had 108 individuals who donated paired CSF and plasma. And they had a routine diagnostic CSF, and then they had paired CSF and plasma run, and we tested phospho-tau217, as well as a number of other promising biomarkers, neurofilament light, GFAP, total tau, and p-tau181. And we looked at the diagnostic performance, and I'll really take you to the headline here of this is phospho-tau217, and you can see those who are amyloid beta positive, so those who have Alzheimer's disease, have almost a fourfold difference. So really this is very compelling evidence that even in a real-world clinical sample that isn't research, these blood biomarkers do work. Now the problem with this initial study was we used the MSD platform. That's a manual platform, so actually integrating a manual platform with manual assays into everyday clinical practice, that's not going to be feasible. So I'm going to fast forward a little bit to the next study we did, which is the LumiPulse. So that's an automated assay, so there's better chance of having reproducibility, so from batch to batch that you'll get the same results. And again, we saw really striking differences. So you can see in the amyloid beta positive here, there's a very significant fold difference between those who are amyloid beta negative and positive, and in AUCs, so that's a diagnostic accuracy of 9.92, so very strikingly positive results. And then what we looked at was this new proposal, where how can you actually integrate these blood biomarkers into real-world practice? And rather than having one cut point of saying somebody's positive or negative, there's a move now to have two thresholds, the top one with a high specificity and a lower one with high sensitivity, so then you can rule in and rule out appropriately. And with having a 95% specificity and 95% sensitivity, you can actually only need to perform 20 to 30% will require LPs. So 70% of people that would have required a lumbar puncture could actually be diagnosed with a blood test. So they could be confidently rule in Alzheimer's pathology or confidently rule out. And this is really valuable because it can mean that more people can access a biological diagnosis. And we've seen from earlier how important that is for somebody's disease course. What is it also important for? So it's a platform for future funding. So if you have a biobank, it allows you to set up other research initiatives. So something that Vanessa mentioned with the asset map is we're very fortunate in Tallaght that we have the National Intellectual Disability Memory Service. And that's a cohort where we see people with Down syndrome and people with intellectual disability. But they're a cohort that have often been excluded from research and haven't had as many opportunities to participate in biomarker programs. But because we had the infrastructure of a biobank, we were then able to move forward and use the biobank to support further funding applications. And that led to HRB funding the BioMind study. So this is a study looking at blood sampling in Down syndrome, trying to figure out the diagnostic validity of blood biomarkers, the impact of biological variations. So do seizures, for instance, impact on blood test levels? How stable are these blood tests? Is the retest reliability there? So if I test them today and I test them tomorrow, would I get the same result? Would I consistently screen in or screen out for clinical trials? And also, I guess, as we move forward towards prevention, an understanding of biomarker dynamics is going to be really important, particularly in genetic populations like Down syndrome, because actually if it's before we see cognitive change, then we need endpoints that are validated, and they're going to need to be biological endpoints like blood biomarkers. And that's where biobanks are really important. What I think is probably most exciting is how biobanks support clinical translation. So you saw earlier all the work we've done with phospho-tau 217, and that then allowed us to put in a funding application for the Consultant Innovation Fund to actually purchase a LumiPulse platform for our laboratory, and we're now in the process of using our biobank samples to validate the assay setup so that we can start clinically rolling out this test. So actually, the biobank has allowed us to prove, okay, this test is valid, it's reliable, and now it's informing the validation of the test at a clinical level so that we can roll it across our service. So really, biobanks are at the cutting edge of driving both discovery research, like the BioMind study, but driving clinical translation. So what it opens up other research opportunities, and what I'd like to talk about now is the all-island biobanks. We saw Bernadette earlier, and Ruth has been in our audience today, and they've been key partners along with DRNI in setting up an all-island biobank. And again, that has many potential opportunities. It can advance understanding of disease biology, enable biomarker discovery and validation, support research translation, and I really hope facilitate collaboration that will ultimately generate public health and policy improvements. So how is the all-island biobank going to work? What are these grand lofty ambitions actually built upon? So basically, there's a shared foundation for dementia research. So the plan is that we'll have a minimum common data set, so that even though centres are working differently, if we have the same common data set, we can pool the same data and then have a more powerful study. It also has similar sample collection and processing so that we know that there's consistency across sites. And this allows us to build infrastructure. So even though we don't have a common one site repository of samples, we know that the same samples and the same data is collected across site. And I think this is really important because it opens up collaboration while also allowing different sites to own their own data and have the PIs at the various sites to have control of their own data. So it's a really powerful potential model that will allow us to drive translation, and also it's important as we think about representativeness and equity of access to research opportunities and translation, that we try and get opportunities not only at single sites but across multiple sites. So this is the idea, and I'm hoping that today is kind of a launch point of it. We can talk about sites who'd like to participate, but also opportunities that such a biobank could be used to collaborate with other dementia researchers so that can be a platform for funding applications, because this is a really exciting opportunity. These are really valuable data sets, and as Mara has shown, you know, the clinical data and the translation into clinical practice is something that's really important, and I think, you know, the biobanks are a critical step for this. So the future, biomarker-supported diagnosis and molecular-specific therapies, I think that is the way we're going to go, and that is the future. But to get there, we need biobanks, we need collaboration. So that's my call out for today. Sorry, I've gone a bit cork now and spoken at 100 miles an hour, but I know we're running behind time, so maybe that's in the back of my mind. Any questions?